Okay, excellent. Hi, everyone. Thanks so much for joining today. And thank you to Alex and Anita and the CMS organizers for a fantastic lineup of speakers this year. I very much enjoyed the keynote talk by Casey this morning. It's a great start to the meeting, and I'm looking forward to hearing about all the progress and perspectives during the sessions. Today, I'll tell you about efforts we have ongoing, mostly in the area of cell culture media, but I'll also touch on some broader considerations for the industry. So let's see if I can advance. There we go. Okay, first a quick look at our company, Merck KGaA, Darmstadt, Germany. It's a 350-year-old company that is still guided by, I believe, the 13th generation of the Merck family. Globally, we are Merck, but in North America, we primarily are known by our three divisions, EMD Serono, Millipore Sigma, and EMD Performance Materials. Our company has four main guiding principles to creating a better world and improve on societal challenges. The first of these, tackling an ever-increasing scarcity of resources, is a main motivating factor for our involvement in the cultured meat industry. Our initiative in this area is driven by our innovation engine, a group called Strategy and Transformation. And coupled with our three divisions, we additionally look to create a connected world, advance healthcare, and scientific discovery and translation. Through our skills and resources, especially as a leading biotech company, we aim to accelerate the cultured meat industry. And this is through developing and providing enabling technologies, all the way from R&D up through manufacturing. We do see that this industry faces some challenges that I'd like to touch upon, but please don't mistake these challenges for pessimism. Rather, these are open areas to innovate and to collaborate to really help push things forward for cultured meat. Over the last few years, we've been distilling feedback from technology experts and key opinion leaders across the cultured meat field and see some main topics rise to the surface. First, of course, costs. Currently available materials for bioprocessing won't allow us to meet price parity with conventional meats. These are tools and materials that were designed for a different industry. And although there is a lot we can learn from biopharma, we'll need to find ways to drive down costs. Then scale up. The sheer magnitude of biomass that needs to be generated likely can't be achieved by current bioreactor designs, at least not with the cells as we know them today. And I really loved Casey's rowboat versus tanker analogy. I think that absolutely drives home the point that we're talking about a lot of cells. And the cells, media, and reactors, they'll all need to evolve together to push this level of biomass generation that we'll need for this industry. Also, we need to be looking up through the raw material supply chain to ensure we don't hit a rate limiting material as we scale up. Scaffolding is perhaps a bit of a longer term play, but we'll need to understand not only how to structure these edible materials, but also how the cells interact with the scaffold. You know, for example, can we take advantage of the scaffold to provide key nutrients or signals that the cells may need? Then regarding data and analytics, this is a nascent industry with key learnings that are focused on developing a manufacturing process and supply chain to support commercialization. Those key learnings are just now beginning. It's a great time to start with advanced analytics and data modeling to accelerate our understanding of cell growth and gain a better just process control and inform better bioreactor designs. Then, of course, there's regulatory. It's still not completely clear what requirements and governance these products will have, but FDA and USDA are engaged, and my colleague Luke Rochelle will share his thoughts on this on Wednesday, so be sure to tune in. Now, looking across this highly simplified representation of the cultured meat manufacturing process, you can see where the four main technologies come into play: the cells, the media, the bioreactors, and scaffolds. But of course, these technologies interact with each other in how well any given process runs. And we need to understand their technical interdependencies and the impact that progress in one area will have on another. For example, although media formulation and scaffolding appear further away from each other on this process map, we know from experience that media developed on one surface modality can completely fail when the same cells are transitioned to another surface. And another example I'll take from Biopharma, a culture medium that pushes high monoclonal antibody titer isn't likely going to be selected over another formulation that achieves similar productivity if it fouls the downstream membrane devices used for purification. So these technologies really do need to be developed and tested across the process to ensure that they work well together. And advancements in one technology area can shape the needs to advance in another. For example, cell line development may ease the reliance on growth factors in the culture medium. Similarly, if cell fate signals can be incorporated into scaffolding, this could again reduce the need to put those factors into the media formulation. And because this talk is focused on media, I'll just highlight a few challenges in this area. The industry is developing a wide range of foods from different species and cell types, and each of those cells will have different media requirements for supporting robust proliferation and differentiation. Cost and volumes per batch and the different culture modes, for example, perfusion versus fed batch, and we'll need to push very high cell densities. So ideally, formulations will reduce, for example, metabolite buildup. What also may help here are ways to filter out ammonium and lactate to facilitate media recirculation. So components need to be stable over the course of the process as well. And lastly, we need raw materials and manufacturing facilities that meet the regulatory standards appropriate for cell-based foods. And again, Luke will get into this in his talk later in the week. Okay, so some more specifics about media. First, I'd like to dispel a little bit of a misnomer. So Essential 8 has much more than just 8 components. It actually has 59. But considering that DMEM was described over 60 years ago, and other classical formulations like Ham's F12 were published just shortly thereafter, maybe it's not surprising that we see basal media as a single component. Ham's F12 was actually originally designed for CHO cells, and we know that since then, both the CHO cells and the media used for CHO-based monoclonal antibody production have greatly evolved. A rough breakdown of the media component categories is listed here. From the salts, sugars, and amino acids to vitamins and lipids, these components cover a wide range of concentrations over orders of magnitude. So you can imagine trace contaminants in a high concentration material like glucose, or even slight concentration deviations in the trace elements could really impact performance of the media. So there are some best practices we can apply for media development and manufacturing. Firstly, well-characterized raw materials with low lot-to-lot variability. And I don't necessarily think this excludes things like hydrolysates. Obviously, if you know exactly what's in your media, you have better control and consistency, but we may be able to manage undefined supplements through testing and potentially reprocessing. You also want a secure supply chain, especially as processes scale up. Of course, the raw materials need to meet high performance standards for the cells to maintain their critical quality attributes and meet regulatory requirements with the supporting documentation in place. So to this end, and also understanding the wide diversity of cell types and species this industry will use to create a vast range of products, we aim to bring forward a toolbox of raw materials for media development that's really tailored to cultured meat and seafood. We're leveraging the diversity of experts we have in cell culture media and bioprocessing, from Lenexa, Kansas to Shanghai, China, from very well-established CHO-based processes to novel therapeutic modalities, including cell and gene therapies. Kim Schrag's lab in Lenexa has been doing the QM7 experiments I'll present in a moment, and Jochen Sieg in Darmstadt has been driving the cost of goods project I'll touch on later as well. And of course, collaboration with our customers and cell-based meat experts such as yourselves is absolutely critical for us to develop the right set of tools. We're also leveraging a large proprietary media panel that's been established over the years across a wide range of species, cell types, and applications. This, coupled with our cell culture knowledge, puts us in a good place for media development and optimization, though for sure we're working on building this panel out for cell types not as common in research in biopharma, such as marine species. So with this large diversity set of panel media and raw materials, there are a few experimental designs we can start with. For example, doing mixture designs for media blending studies, or DOEs for specific components we want to query and gain understanding of how these components interact. Our standard culture formats include 96-well plates, both for adherent culture and suspension. And these interface really well with analytical instruments such as flow cytometers and plate readers. With strong enough data sets, we can do a range of statistical analysis to identify, for example, the major components influencing any given measured attribute. We can also layer in spent media analysis to identify components that may be limiting to carrying capacity of the media or identify metabolite buildup. And all together, this drives toward a media formulation that balances all the desired end goals, not only of the cells, but also with regard to cost and media complexity. I mentioned we're looking to build out our experience and a panel for cells more relevant to cultured meat industry, and found that QM7 cells are a good place to start. This is a quail myoblast cell line available from ATCC that expresses lineage-specific markers, both during the proliferation stage, shown here in the top set of images, as well as during the differentiation stage upon myotube formation, that's shown in the bottom set of images. So alpha-tubulin and PAX7 can be detected in myoblasts, whereas myosin heavy chain and desmin are apparent in the myotubes. The standard media for QM7 cells, though, includes classical basal formulation, that's M199, and it's supplemented with fetal bovine serum and tryptose phosphate broth. And in these pictures, you can see how the cells look under bright fields during proliferation myoblast stage on the left, and during the differentiating myotube stage on the right. So we initiated a media development project to eliminate serum from the media, transition the cells to suspension culture, at least for the proliferation stage, and also develop a formulation that would retain the cell's differentiation potential when they were put back into adherent culture. In the first step, we wanted to identify a basal medium that would at least support lower serum concentrations. One of the formulations we tested was actually one of our catalog media, KGaA1, that was originally developed for an avian species, so we thought that was a good place to start. On the left bar graph, you can see how the doubling time normalized to that of the control medium, but with reduced serum, so now with 2.5% serum. I should note that KGaA1 does contain hydrolysates, and increasing the amount of calcium also helped this cell line in adherent culture as we reduce serum. And at this point, we also tested a chemically defined prototype that's related to KGaA1 formulation, but it did not perform well under these conditions. So using the modified KGaA1 as a basal medium, we did a supplement DOE, or design of experiments, to eliminate the serum altogether. This was a basic 6-factor 2-level study, and we were able to identify a few conditions that sustained culture over multiple passages. We also used this study to compare a few different culture formats since we were still setting up our assays. There were a few conditions that grew well over multiple passages, as seen in the bar graph here on the right. We then transitioned the cells in these top formulations to a few modifications thereof For example, now going back to the reduced calcium levels, we transitioned these to suspension culture. The sawtooth growth curves over multiple passages here on the left shows that a few different formulations were able to support suspension culture quite well. And number 23-2 was chosen for direct adaptation to suspension culture that you can see here on the right. Although growth did markedly slow down between passages about 3 and 7, by passage 8, the cells start to normalize again. And over the course of this suspension adaptation, the viability never dipped below 80%. From there, we wanted to further optimize the formulation and did a second round of DOE. Again, 6 factors, but at 3 levels now. And we added some extra controls and examined a few additional factors, just 1 factor at a time. So now with more conditions, we utilized TPP tubes, which we found to work quite nicely with these cells, actually. We immediately discovered that one of the factors in our study is critical to maintaining growth of these cells. So although we generally want to avoid loss of a set of DOE conditions, these are early exploratory experiments here for us, and we did identify this critical factor. Basically, we're trying to define a starting serum-free formulation from which we can then enter that media development paradigm I set out a few slides ago. So we expanded and banked cells in the top conditions here in the right bar graph, and those grew really well after the initial adaptation period. During the study, we also checked whether any of the DOE conditions supported attachment and differentiation of the cells when they were put back into planar culture after being expanded, in suspension. And perhaps not surprisingly, the suspension formulations we were developing generally did not support reattachment. After all, they were designed for suspension and proliferation. But importantly, we did demonstrate that the cells at least retained the ability to reattach and differentiate by putting them back in the FBS control medium as well. And some of the conditions also formed myotubes when the FBS was further reduced, as seen here in the right pictures for condition number 9. Up to this point, we've been working with GROW1 that has hydrolysates. But now that we have a promising serum-free supplement, we wanted to go back and check whether our chemically defined prototype worked. And sure enough, when supplemented with the number 9, we see strong cell growth over multiple passages, as shown in this sawtooth graph. But we also want to know what would happen if we let the cultures go to crash. So at each passage, the old flasks were left to continue culture for a few days. And we saw that the chemically defined medium plus supplement number 9 has a much higher carrying capacity. In other words, the cultures were able to reach much higher cell densities. And this is as far, unfortunately, as the QM7 story goes for now. We're still working on it. Next steps will be to move into formulation optimization for the proliferation medium and also start developing the differentiation prototype medium, ideally using the same basal as the proliferation, but just with a different supplement. Okay, I have 2 more things that I'd like to tell you about today. The first being our efforts around cell culture media raw materials. We are taking a comprehensive look at all the media components we're interested in using for cultured meat processes. And we're doing a risk assessment on availability, cost, and criticality to this application. In other words, how often does any given component appear in clean meat relevant media that we have in our library? Right now, we're pulling on mammalian formulations, avian formulations, and insect formulations. So in this heat map on the right, highest risk components are the large circles in the upper right quadrant in red. We're also doing an item-by-item regulatory assessment, but again, my colleague Luke will speak to this in a later session. In parallel, we're identifying food-grade sources of these raw materials and setting up our testing paradigms. The end goal is to ensure that these raw materials are suitable for this application, cell-based meat production. Our applications testing in R&D is coming in two main waves. The first is to test a distinct set of components that we see as the highest risk. For example, glucose and NaCl, since they're in higher concentrations in the medium. For this, we're leveraging our pilot scale dry powder media manufacturing facilities to make backbone formulations that are deficient in the set of components that we're querying. This really eases experimental setup, as just a few components have to be added back to complete the medium. And we can test many vendors over many lots using that same backbone dry powder media. At this point, we'd really like to understand how, for example, adding table salt might impact mammalian cell culture versus biopharm grade NaCl, especially if trace contaminants are present, or there are differences between these preparations of raw materials. For the second testing paradigm, we're moving to the first principles approach. That is, rather than using dry powder media backbones, we'll formulate all of the individual raw materials in concentrated stocks, then use liquid handlers to create any set of pH and osmo-matched formulations we want on the fly. This will allow us to run more complex DOEs, generate fit models, and do factor profiling to get even better and optimized formulations and get there faster. And because there absolutely are vendor-to-vendor differences in any given raw material, we'd like to get those locked as soon as possible. And this is all toward creating a toolbox of food-grade raw materials from which media for, again, any cell type or process can be developed. And lastly, a few thoughts about scale-up. So there are two approaches that can be taken as a new process evolves. The dish up or bioreactor down approaches. In other words, should the focus be on getting the cell biology and the application to work on the bench, then design a manufacturing process around that? Or should one start with the endpoint in mind and work back and down from there? I might argue that organizations that successfully bring experts from both sides together will be the best positioned. Right now, the path isn't quite clear, and estimated current production costs are very wide and don't yet meet that of conventional meats, though some are inching closer and closer to this, which is really promising. And in order to understand a bit better where we are using today's technologies, we partnered with Biopharma Services to generate a cost of goods analysis for a scale-up process based on their BioSolve model. The main goal was to understand the differences between three potential reactor modalities for the biomass generation phase: single-cell suspension in stir tanks, microcarrier-based culture of adherent-dependent cells in stir tanks, and the third, hollow fiber culture. We also wanted to understand, if we pull the lever on media cost and productivity, how much would that impact the overall cost of goods? And also, what would be the breakdown between the main cost buckets, capital, materials, labor, etc. Now, this model has many assumptions for sure. And the utility of any given model is certainly dependent on these assumptions. A couple of those key assumptions are here. Again, this is using today's technologies, today's assumptions around cell doubling times, and also maximum cell densities for mammalian processes. We also varied the bioreactor mode for the proliferation stage, as I mentioned, and also looked at the media costs that were estimated based on the current price of FBS. The differentiation stage and harvest, though, those were kept constant in this model, and we based it on a hypothetical fixed bed process. Importantly, the final product yield was only based on cell biomass, and it does not include any weight contributed by the scaffold. So overall, we see this as a very conservative model. And if anyone is interested in discussing the model in more detail offline, please do reach out. We really still are playing with it, running different scenarios and understanding it. But we see some trends emerging from the model. Suspension culture mode achieved the lowest cost of goods. So the bar graph here shows the COGS per gram meat for 3 biomass modes, the suspension, the microcarrier, and the hollow fiber, at 3 different process yields and 2 different media costs. At the 1 ton per year throughput, which is graphed here, suspension has the lowest upfront capital investment. And in this mode, one can drive down the costs the most based on lower media price and driving higher cell densities. The graph looks a bit different at the highest throughput. That was based on scaling up and out, most notable to 12 bioreactors running in parallel or staggered in a single facility. And for the suspension mode, that's 12 25,000 liter bioreactors. And the bottom right table here denotes the differences in the yearly yield. So again, this is maximum throughput for each of these modalities, with suspension achieving over 300 tons per year, whereas the other process modes are less than 100 tons each. This difference is also reflected in the installed capital differences as well. So although installed capital is much higher for the max throughput suspension scenario here on the left, you're running much higher capacity reactors, and therefore you have a higher yearly throughput. So the cost of goods per gram of the final product are much lower. Now, this is still showing $1,000 per kilogram of meat. Again, very conservative model, lots of assumptions, but we should consider these trends and let them help guide us as we look to scale up and design new bioreactors, etc. So in the best case scenario for this model, the suspension mode at lowest media costs and highest cell densities, we also want to understand where's the money going. And we see some further areas for improvement. In the red, there's still over a quarter of the costs associated with materials and media, especially during the differentiation phase. And remember, that was fixed in this model. And also utilities start taking up more of the pie, as noted in the green wedge. But capital accounts for almost half of this scenario. So there's also plenty of room for improvement on the bioreactor technology side as well. And this could go in many directions, but I would love to see, for example, some continuous processing technologies emerging here. And to this end, on exploring new bioreactor designs, we have been running an innovation research call. One of the target areas this year is bioreactor design specifically for cultured meat. We're in the process evaluating the 64 applicants. Some very creative proposals have been made. The top teams will present their final pitches in November, so stay tuned for award announcements thereafter. We're really excited about being able to support research in this area of cultured meat industry as well. So with that, just a few closing thoughts. Having a food-grade toolbox of cell culture media raw materials and formulation development strategies will serve the broad range of species and cell types being used now. And as the industry advances and evolves, so too will this toolbox and these strategies. And although media performance and price are major drivers, we also do need to advance the other technology areas if the full potential of cell-based meat that can compete with conventional products is going to be realized. So thanks so much for your attention, and I'm happy to take questions. Very much looking forward to the further presentations and panel discussions. Thank you.
Thanks so much, Aletta. That was a really, really illuminating presentation. Thanks also so much for all the work that you showed that you guys have done on so many super critical areas of research in this field across the board. I especially appreciated this QM7 case study. I think it really helps to demystify the process of, we often like sort of wave our hands and say, media development. And I think that this was super incredibly useful to understand what really goes into that and what that means, and the time and effort that it takes to get to sort of a working prototype.
For sure.
So we have quite a few questions coming in here. The first is connected to the cost of media production. So this is from Tariq. The question is, what is the expected blended cost curve of a suitable, so a high volume potential complete medium to produce a product like beef? And if today the cost is 100% for the medium, can you sort of attempt to project the cost in short term, few years, long term, couple decades? And if it goes up or down, what are assumptions or drivers for those cost changes?
Sure. So I'll speak to the general cost of the medium. We see that it does need to be obviously driven way down. Right now, the complex media formulations for cells like mesenchymal stromal cells that require a lot of mitogenic input are in the hundreds of dollars per liter. So we need to drive that down in even the shorter term to the tens of dollars per liter. And then I'm looking a little bit further out as companies start to scale up their manufacturing, into the single digits, really.
Moving away from the cost for a second and towards more the sort of safety aspect and the risk assessment you were describing. Great question from Elliot Swartz. Do you have recommendations or predictions for methods of media sterilization in future facilities that don't harm, for example, ingredients that are not super stable at high temperatures?
Yeah, that's a really good one. So it's definitely, sterility not only of the media, but just sterility management of the whole process is going to be important, because absolutely you want to get away from antibiotics, but these are going to be big scalable processes with a lot of cells that cannot outcompete, other things that could contaminate the process, like bacteria or yeast. So for sure, general sterility management is a concern. There are techniques such as obviously filtration. So for sure, 0.1 micron, 0.2 micron filters, those can be greatly scaled up. There's also some potential to gamma irradiate powders, right? So that is another thought. There could be some utility around high temperature short time, but we'll see if that's sufficient. I have a little bit of doubt there. And then, I think media formulation, having components that are stable, and it's not just for the sterilization, but over the course of a process. We know that growth factors can be quite labile. So we're being able to replace those with things that are more stable, for both the process and the sterilization, I think will be helpful.
Definitely. Thanks a lot. Another question that's less about the contamination with, for example, contaminants, but more about sort of cross-contamination with other products. This is coming from Brian, and the question is, will Merck manufacture food-grade dry powder products using the same equipment that is used for pharma grade? And how do you avoid sort of cross-contamination between products of different quality scales?
It's a really good question, because for sure our cell culture media manufacturing facilities right now are biopharma grade. So we are working with regulatory agencies to gain their perspectives on what will need to be done to keep both processes safe for both applications, right? So we certainly don't want to risk either those manufacturing facilities or the end applications. So we are working with agencies to understand what the requirements are, what the risk assessments need to be. Luke is going to get a bit more into detail on that later, but the questions are, do we need to build out completely separate facilities, or can there be some co-localization of those? Those questions are still being asked, but for sure we will use the full knowledge and full clarity as we push things forward on the food grade side.
Thanks so much. So responding to your answer earlier that filtration systems are a great way to sort of ensure that you are avoiding contamination. Another question from Brian: is Merck working to radically increase the scale and decrease the cost of media filtration systems that can prevent virus entry in media and resulting viral contamination of a bioreactor?
Yeah, I think that for sure that's another area for innovation, and we have to think about new ways to create membrane, or maybe it's not even flat sheet membrane. Maybe it's another modality for the sterile filtration. Maybe we can capture some of those adventitious agents. So that is something that we have our eyes on. And we'll see what happens down the road. But yeah, absolutely, a lot of work needs to happen there. Again, those are made for pharma processes right now, and we have to see, as these technologies evolve together, will we need a pharma grade 0.1 micron filter? Maybe yes, maybe no. Hopefully no, and we'll have to innovate more cost-effective ways to manage that.
Definitely identifying sort of bottleneck parts of the bioprocess where this is necessary, but other places where it's not. I think it's all a learning game here.
Exactly.
But I really like the language that you use to describe challenges as more opportunities or open areas of innovation. I think it's a really good way to think of it.
Yeah, and we see this is not a matter of if, it's a matter of when, right? And I think that there are a lot of other industries that have been like that. This challenges seem a little bit insurmountable at first, but together, having conferences like this where we can have open dialogue and really network and start to brainstorm some of these solutions, because we don't have all the answers right now, for sure. We need to innovate and get there, but we'll get there.
Absolutely. So question from, I think it's pronounced Eon. How close to the market are your serum-free proliferation media, and do you think they'll be commercially available in the foreseeable future? Have they been validated in other cell cultures or in other cell lines other than the avian ones?
Right. So we have a vast portfolio right now that's on the market for research use and biopharma use. So I would say for this industry, there's still research use, because we are not there at the point where we have the food grades, raw materials, and manufacturing facilities worked out yet. That's exactly what we're working on now. So for sure, we have relevant basal media formulations for research use. We also, much of our activity in the media field is actually custom formulation. And, CHO has been around for decades. And there is still, that platform is still evolving. The media formulations are still evolving. And each process, each product is unique. And so are the media formulations that support those. So this will continue to evolve for sure. Right now, we're at the research stage.
Yeah, and great that you say this, because Merck's supporting early stage research in this area, I think, is incredibly vital as well.
And that's why we're trying to also gain our knowledge with across a couple of different species. Again, screening our portfolio media so that when someone comes to us and say, hey, we're culturing these cells, do you have any recommendations, that we can at least have a starting point.
Great. Great question from Kai, who's wondering if there might be a sweet spot between assuring batch-to-batch variation of media components and cost, or if the most defined formulation will be the most efficient in the mid to long term.
Yeah, I know, and it's, we're so torn because on one hand, just from experience, knowing what's in your media is really helpful, right? So it's very challenging, especially if you have a vendor change, or you have a supply chain issue with an undefined component. It's really hard to find a suitable replacement because all of these undefined components are different. I mean, to the point where people are screening many, many different lots, reserving huge lots of FBS. It becomes a challenge in biopharma worlds, and I don't see that challenge will be different in the cultured meat world. We have to really think about that supply chain, because on the other hand, I think using hydrolysates or I don't want to say byproducts from other food processes, I think will really help this industry to drive down costs, and also resonates very well with sustainability. So perhaps it's just, as I mentioned earlier, a matter of how those are tested. Perhaps there's a little bit of reprocessing that can happen to bring them to the performance standard that they need to be, to find that happy medium between cost to performance, and whether it's defined or undefined.
Yeah, definitely. Another great question from Elliot from GFI, which is interesting for everyone. Are you going to publish your COGS model or develop an open access web-based tool from it?
Those are two very good suggestions. And I think that my colleague, there are some more conferences coming up, and my colleague Jochen Ziek, who's really been driving this effort, maybe there's an opportunity for him to present a little bit more thoroughly on this analysis. So stay tuned for that. A great suggestion, Elliot, for making this a bit more open and publishing on it. We certainly want to share the information, and it also would be great to get people's perspective. So anyone who's interested and also has experience in analyzing these kinds of models, maybe scrutinizing the assumptions, definitely open to having those conversations.
Yeah, it would be an incredible tool for people. So a question from Vince, which is, do you plan to seek regulatory status, for example, the generally recognized as safe designation, for individual media ingredients such as growth factors for the purpose of cell-cultured food production?
Yeah, so Luke is going to get into this. So he is the expert. I'm learning a ton from him, but actively engage with the regulatory agencies to understand what the path forward is, because we have identified, although many of the raw materials for media seem to be clear and already use in the food industry, there are some that we've identified that are watch-outs, and we just need to understand better. There's a couple of approaches there. From the R&D side, I'm thinking about how can we formulate them out? Are there suitable alternatives? Maybe if we take this set and we find some alternatives, we can provide the cells with the same nutrients that they need, but using materials or supplements that are already in use in the food industry. So from the R&D side, that's what we're kind of taking that approach. How do we just avoid using them altogether? And then from the regulatory side, Luke will talk about the efforts toward defining things as generally regarded as safe and food grade and all of that.
And what are some of these watch-outs that you have in mind?
So a really obvious one is phenol red. We need to get rid of that. Right? Just stop using it. So get rid of the phenol red. There are some others that are used in bioprocess. For example, poloxamer. That's a shear protectant. It'd be interesting to see what we do with shear protectants as things scale up. Are there others that we can identify? Poloxamer is a complex one because it has to be only certain molecular weight species are really effective and that are tolerated by the cells. Are there alternatives? Maybe some cheap bulk protein that can be put in that can serve the same or similar effect. So yeah, there's a handful. I'd say 10 to 20 that we need to dig a little deeper.
Open areas of innovation.
Absolutely.
A great question from Michael about going back to the cell universe here. How much of an issue or a gap is there when you move from 2D cell culture systems to suspension culture when you're creating these formulations?
Yes. So I'll give another example from cell therapies in my previous team. We've been developing media for mesenchymal stromal cells and had a formulation that was so far superior in planar culture. It was developed in planar culture, and the identity markers were spot on. The differentiation was beautiful. It was just so good. And when we transitioned that to microcarriers, it was like a complete failure. I think 5% of the cells attached. It just was so deficient in something that the cells needed to make that transition. So that can be a challenge. And I think that, and I know that folks in this industry are doing this, bringing their critical quality attributes that will come to play later on, in the final product, in the process, and bringing those really early in their development process, developing high throughput assays, ideally in a perfect world that are predictive, right, of what that final product is going to be and what that final performance is going to be. Bringing that measurement and tracking as early in your process and media development as possible is going to be really critical here. And I get that for some cell types, this is challenging because there just aren't the research tools. There aren't a lot of antibodies available for some of these marine species. So that's absolutely a limiting part of it too. But yeah, we see that can be a challenge. But again, if you develop your high throughput assays such that you address that early on, I think that really helps ease it.
Yeah, definitely. It's also difficult, I think, to make this sort of mental connection between the mechanical needs and the chemical needs of these cells. Yep. We can kind of view it in two sort of one-dimensional manner.
Exactly.
So very specific question about hollow fiber bioreactors used in the system that you're describing from Alex. For this hollow fiber bioreactor technology that you described in the media optimization experiments, what is the approximate diameter of the rods in these types of systems?
Ooh,
super specific.
That's really specific. So I can tell you that analysis was based on currently available technologies. So using hollow fibers that are available now, I don't know exactly what the inner diameter of those is, but it's using today's technology. So that information should be pretty straightforward to find. Just follow up with me.
And the media tests that you showed us, they were done on this. So in one case, the quail myoblast cell line. Have you also tested media in primary cultures?
Yeah, we are working on primary cultures as well. And, absolutely, it's a different set of considerations, especially around the suspension adaptation. So it is more challenging. The quail cell study is really to kind of vet our approaches and get our assays in place, test, our panel media, and have that starting point. But primary cell cultures are going to be more challenging, and we're seeing that as well. And also, I think, though, that it depends on how the cells are selected and how they're originally isolated. We've worked with a couple of different primary cell cultures across different species, and they're not all the same. Some of them really, after a couple of passages, peter out, and others, based on, I'm assuming, the way they were isolated and selected, do quite well. And so I think it's certainly possible to refine those isolation techniques along with refining the medium and to drive the higher population doublings. So then, that might be a different story. But yep, we've tried the primaries. They are more challenging, but and it's an area where I think that Casey got the question about, where would you like to see more research? And I think understanding some of that basic biology for these clean meat specific species is really an area that we need to also focus on.
Yeah, absolutely. And again, this more holistic view of the mechanical properties of where they get seeded immediately after they're isolated, that is definitely going to change the population that survives.
Exactly.
Everything in synthesis.
Yes.
Great question from Suzanne. What is your opinion about how inexpensive media has to become to enable mass meat production to be competitive with current meat prices? Big question.
That is a big question. And the thing with that question is that it also depends on, again, the carrying capacity. So it's not just the cost of the media, it's how dense of a culture will that media support. Also, can we develop technologies that, again, can scrub out ammonium and lactate or some of these metabolites to allow that media to be recirculated and really use up all of the components? Can we then envision really concentrated feeds that get injected so we minimize the overall volume of media that's being used? So I find that it's a little bit challenging question, but like I said before, we're definitely seeing a need to drive, into the tens of dollars per liter and down into the single digits to really facilitate scale up.
Yeah, of course, it's always a really, really big and difficult question to answer with so many sort of moving parts connected to also economics and supply chain that you cannot really predict from here. But
exactly.
Thanks for the answer, definitely. So coming up in the last few minutes here, we'll probably just take a couple more questions. Really interesting one from Laura Amundsen. Do you see any space for traditional animal agriculture nutrition companies in helping to formulate culture medias? We formulate diets for animals to meet the basic biologic needs of animals and to be the most efficient they can be. So Laura writes that I could see a benefit of using top quality nutritional ingredients, for example, vitamins and minerals, which are available in different forms. So in bioavailable forms from companies like these in media formulations. What do you think about that potential overlap?
Absolutely, yes. I mean, for sure, our media have the vitamins and the minerals and the amino acids. Again, as we're thinking about, really bulking the protein, we want more amino acids. I think that taking a nutritional approach is a nice one, and understanding how some of these different diets may impact the taste even and texture of the final meat. I think that could really become interesting and help guide not just the media formulation, but also the final product and maybe even the scaffold, right? So I think the scaffold is a nice opportunity to introduce or reintroduce some of the nutritional qualities as well. So yeah, I love that idea.
Yeah, definitely. The sort of bioavailability of nutrients in both the media and the scaffolding is going to be the sort of most relevant part for the final product. By mass, that's what most of it's going to be. Another great question from Brian, and I think we probably will have this the last one for the session. So Casey Carswell's presentation suggested that medium demand for clean meat will be many orders of magnitude larger than our current for biopharma. Yes. A single clean meat plant might use more medium than all of current biopharma, very legitimately. What proof of market success will Merck need to trigger your building the huge medium production plant that will be required?
Yeah, I think that it's going to be a stepwise approach, right? So we have a short-term, a midterm, and a long-term vision, and would like to grow with the field for sure. And it's not just about the media manufacturing capacity, to Casey's point, it's also about the supply chain of those raw materials. So I'm actually not as worried about the cell culture media manufacturing facility itself. It's the materials to ensure that we can source enough of those raw materials. That's something that we need to look way up at the supply chain and make sure that even Merck KGaA, that our suppliers also scale and are motivated to scale with this industry as well. So yeah, we'll evolve with it for sure. And that's how it always is, right? You have to have some successes, and then with successes, you get further confidence, more funding, and then you expand from there.
Great. Well, thank you so much, Aletta, for your very, very thoughtful answers and a really great presentation. Like I said, I think it was very helpful for everybody to hear how this process works and to see the support that's coming from your team and Merck.
Thank you. Yeah, we're really excited to support the field, and I'm happy to participate and can't wait to see the rest of the panel talks and presentations. Thank you, guys.
Me as well. Thanks so much.